Hey everybody, today is April 16th. I'm Mr. Sheridan. This is Zoology. Hope everybody's having a great day. So today we are going to transition from talking about ruminant digestive systems to monogastric digestive systems. So if you will, please jump into your Google Classroom, and under week 32, you have assignment number 68 is lesson 4 notes on the monogastric system. It's going to look like this. You don't necessarily have to fill out this top part if you don't want. Google Classroom does that for us and lets me know what is going on. Oh, I should start with this. Just so everybody understands, if you did not get the emails, the high school is going to be virtual again next week with the hopes of starting again on the 26th of in-person learning. So we will be here online in Zoom next week for zoology, and hopefully we can transition the week after that into doing some labs and talking about digestion that way. But because that's been so delayed, and because we have so many seniors in this class, I've made the decision that we are not going to do an actual live poultry feeding project. We'll do what we can to demonstrate as many things to you as we can. But by the time that we do get back into person, even if it's on the 26th, we only have about 3 weeks until our seniors are gone, and not really fair to have 4 or 5 people doing chores every single day till the end of the year. So I've decided not to do that. For those of you who are coming back and not seniors, we will have that. We're going to do poultry in the fall, like we've done for the last however many years. So we will be doing that. But so we're going to do our best to talk about feed rations and how to create feed rations. We're going to talk more about the nutrition systems in animals, but we're not going to demonstrate that through a live poultry project. So big old bummer, but that's the way that it goes. It's just we're all kind of rolling with the punches right now. So you need to have this document open. We will cover these things, and you will be able to fill this diagram out by the end of class today. So as we get started here, you all should be able to identify what type of system this is, whether it is a ruminant digestive system or a monogastric digestive system. I would assume that by now you can see that there's only one compartment to this stomach, meaning that this is a monogastric digestive system. And that's what we're going to talk about today. Again, I will give the caveat that this presentation was put together by I'm assuming either an undergraduate or a graduate group of students. You'll see some of my pet peeves coming through in this presentation as far as layout design. It drives me crazy, but we're going to put it together and roll through it because there's a bunch of really good information. It's just organized in a little bit PC way. So our objectives today, we're going to evaluate which animals are monogastric. We are going to analyze the components of a monogastric stomach and a monogastric system, digestive system as a whole. We're going to interpret the parts and functions of monogastric digestive systems, and we're going to analyze the enzymes in a monogastric digestive system. That part gets a little technical. There's some cool words that we're going to talk about today, but as a Zoology 2 class, we do need to get into some of that stuff. So what is digestion? Digestion is the process by which large, complex nutrient molecules are broken down into simpler molecules capable of being used by the organism for food. Pretty basic stuff. Through both chemical and mechanical digestion, food is broken down so that we can use it as animals. So a simple stomach. These types of humans, swine, rabbits, chicken, horses all have simple stomachs, which is also known as a monogastric digestive system. You'll notice that this was created in Texas as we got our friend George Strait as our representative of human. You'll also notice, for those of you who are paying attention during the livestock judging portion of our class, that this boar looks a little different than the ideal boar that we were looking for, or even the barrows and market hogs and gilts than we do today. This is kind of what we were looking for about 20 years ago when I was when I was your age, that we were trying to get these pigs as tall as possible and as lean as possible. That was kind of the goal. So this would have been a very good boar in about 2000, that he was super straight, very straight in his shoulder, very steep out of his hip, very straight in his hock, very tight in his rib, shallow bodied, but that's what we were looking for 20 years ago. And in pigs, it's very easy to transition from trying to identify some traits that you like and then move to others. Right now, we're looking for more width, rib shape, depth of body, that sort of thing, along with muscle and structural correctness that would be very different from what that boar is trying to show you there. So this would be another pet peeve of mine, is the transitions. This is ridiculous. So monogastric digestive systems, carnivores and omnivores, meaning that, so basically anything except for an herbivore, they have monogastric digestive systems. And even, I'm trying to think of some herbivores that would. So we're talking about horses and rabbits are going to only eat, are not going to eat meats as well. So we're talking about, there are monogastric digestive systems that are herbivores as well. Monogastric systems have a simple stomach structure that only has one compartment. And some animals with monogastric digestive systems are pigs and rabbits, humans also. This is where you're going to fill out your guide. So I'm going to keep it here for a few minutes, and you should be able to fill out your diagram from this. You'll notice up here, both on yours and here, it's a little tricky to figure out what exactly they're pointing to. I think that one of the things they're pointing to on your diagram is teeth. The other one is either the tongue or the mouth. You decide which one you want to do. Doesn't matter to me much, as long as you identify. And this is pretty simple stuff, that you've got the mouth, tongue, teeth, and the salivary glands that are there in the mouth area inside the head of a pig or any monogastric. What your diagram does a better job of than this one is identifying what the liver actually looks like. The liver is a very large organ, and it covers up the stomach. If you were to cut into the side of either us or a pig, you're going to see the liver first. It's dark red color because it's got lots of iron in it. And you'll notice that large, in fact, we can go back and just take a look at your notes page. So you'll see this number 7 here has both the liver, which is this large organ, and the small accessory organ, which is, you saw from the slide, the gallbladder. So bile, which aids in digestion, aids in breaking down proteins, is stored in the gallbladder. So it's made in the liver, stored in the gallbladder. Those are connected, and then it helps to break down food. The stomach. 6 is going to be the pyloric sphincter. So you can say the pyloric area or region. If we go back here, they don't have that label. Well, if you wanted to call it the duodenum, which is the first part of the stomach, that's fine. But I'm going to actually call it, and that's your, this is what we're talking about down here, is your pyloric sphincter is what they're going to be referring to right here. Then I'm going to call this, we got number 7 was there. Number 8, we can call the pancreas, which is another accessory organ that aids in digestion. Number 9 through 12 gets a little dicey. 12 is obviously either should be the anus. 9, 10, 11 gets a little dicey in here. I'm going to call 9 the small intestine, 10 the large intestine, and 11 the cecum, because it looks like it's kind of halfway in between both. We're going to talk about what the cecum is, what it does, particularly in terms of horse and rabbit digestion, because they both have if not a large, but a functional cecum, where pigs and humans don't really use our cecum for much anything. Does anybody want more time with this diagram? Does anybody need more time to fill out your notes page? Okay, now we're going to move on. All right, so your monogastric stomach, just the stomach itself. So in ruminants, we have 4 different compartments in their stomach. In monogastric stomachs, that one stomach is split into 4 different parts. So we start with the esophageal, which is kind of just the very, very beginning. Doesn't have any glands. It connects the stomach to the esophagus. The cardiac area is right inside that stomach. It's up next to the heart. That's why it's called that. Your mucus secretion is going to start here. You're going to start to get some of that chemical digestion. So you've got to have that mucus in order to protect the stomach from basically eating itself, which is, that wouldn't be good. The fundic part of the stomach is the biggest part. It's the biggest part of the stomach. It's where you have both mechanical, this digestion and churning is going to take place here. That would be mechanical digestion, and mucus secretion, which would be referring to chemical digestion, meaning that you need that mucus secretion to protect the side walls of the stomach from the chemicals that are in the stomach, because they will just eat itself if it wasn't for that mucus. And then you've got the pyloric area, which is the very bottom of the stomach, or the very end of that stomach tract that connects to the small intestines, and also secretes some mucus. So this is what we're looking at here. We have our esophageal area, and then the cardiac layer up here where they have the different types of muscles layered up here. You've got your gastric folds that we saw in the abomasum of a rumen's stomach yesterday. You can see these folds. It's where we saw that kind of mucousy glistening on the lining of that stomach that protects the side walls from the hydrochloric acid and other acidic secretions that are inside the stomach. And then you've got your pyloric area of the stomach, which is connecting to the small intestine. So it's very, very acidic, obviously. The pH drops to 2 to 3.5. To give you some reference, the pH scale goes from 0 to 14, 7 being water, which is neutral. So we're talking about something that's very, very acidic when we're talking in the 2 to 3.5 pH range. So amylase stops working. Amylase breaks down proteins, is another one of those. So we're not having that work in the stomach because the pH is too low, but we need this stomach lining to be able to do that. So we've got to keep this smooth muscle going, very similar to our heart that is also made of smooth muscle because it works so much all the time. Hydrochloric acid is produced, which denatures proteins and prepares them to be broken down, which basically means it starts breaking down the proteins. We're going to talk about a couple of other secretions that the body makes that further breaks down proteins, but you need to know the hydrochloric acid kind of starts that process. It weakens the bonds, the hydrogen bonds that hold proteins together, and therefore they're able to get digested and further broken down, both in the stomach and in the small intestine. Pepsinogen is the inactive form of pepsin. So what the two things that are working together here are pepsinogen and hydrochloric acid. So hydrochloric acid actually it activates pepsinogen in so that it turns into pepsin. And pepsin, which actually breaks down and denatures those proteins even further. So the hydrochloric acid, not only does it start to break down those proteins, but it also activates pepsin, which further breaks down proteins. So it allows us to eat the proteins that we need. Here's my pet peeve, everybody. So when you have a group project, when you're doing a group presentation, for heaven's sake, use the same font and use the same, this is insane, but I'm stealing it from them, so I'm not going to complain too much. Okay, parts and functions of the monogastric mammal digestive system continued. So in our small intestine, so now we've talked about the stomach, the few secretions that are there. It's mostly hydrochloric acid that's doing most of the breaking down, and then it's activating that pepsinogen into pepsin so that it can break down some proteins. But we've got enzymatic digestion and absorption happening in the small intestine, a lot going on here. So we just talked about this a lot in our 7th grade science class, actually. So I imagine that through most of your stuff, and those of you who have taken biology with Miss Ebert, you're going to kind of be familiar with this stuff already, that there are villi in the small intestine that are aiding in absorption, increasing the surface area in your small intestine. So it needs those enzymes to be able to break down those carbs, fats, and proteins into smaller, simpler molecules, meaning that Basically, when you eat a carb, it's a long strand of glucose that's all held together by bonds that keeps it so that you can't absorb it. So it needs these enzymes in your stomach and small intestine to start to break down those bonds so that then it gets broken down. For example, carbs get broken down into simple sugars, which are glucose. And glucose is what our cells actually absorb in order to give us energy. It's the cellular respiration, which is the opposite of photosynthesis. Those two things work together. So we've got to get those things broken down to change them from carbs, proteins, and fats into the simple molecules that our bodies can actually use. So that's where this is all happening. So we've got the function of the small intestine is the digestion of proteins, carbs, and fats, and absorption of those end products that actually our cells are allowed to use. So that villi absorbs those simple molecules and then is allowed to kick it into the bloodstream, which takes it all over our body so our cells can use them. So most of your digestion is going to occur right away in the small intestine, the first part, which is called the duodenum. The jejunum is kind of half and half of digestion and absorption. In the ileum, we're starting to transition into that large intestine cecum area where we're absorbing both the enzymes that were in the digestive systems and the water to aid in hydration. So, oh, and I told you bile broke down proteins, and I apologize. That's not true. Bile actually emulsifies fat, which is one of those things. So we've talked about how pepsin breaks down proteins. Well, bile does the same thing. It emulsifies fat. We're talking about breaking the bonds that holds those molecules together so that they can become small enough that our body can use them. All right, so now that we've gotten through our small intestine, now we're at our cecum, which is in most of us, meaning humans and pigs, it's basically non-functioning. But horses and rabbits have a cecum that's very, very important to their digestive system. They have an enlarged that acts like a rumen. So after some of the normal digestion has taken place in the stomach and small intestine, then a lot of that fiber is still there that hasn't been able to get broken down through the normal process of going through the stomach and small intestine. So it's got to sit there in some of those enzymes and get further broken down and absorbed into the body. And that's what the cecum does. It acts as a fermentation vat that allows those cellulose to get broken down. So, and then we're moving into, after the cecum, we're moving into our large intestine, which is where there's a lot of bacterial activity, which is why you don't eat poop. And water absorption is taking place and waste storage. So here is a list. You do not have to memorize this, but it is really interesting to figure out where these things are coming from. So these are all the enzymes that are aiding in digestion and where they're coming from. You can see that your pancreas is incredibly important because it's dumping a lot of these enzymes into your small intestine, which is where these are, where we're breaking down carbohydrates, lipids, and proteins. So it's got enzymes to do all that stuff. Those are the three major food molecules, carbs, fats, and proteins. You've got to get those things broken up from large strands into smaller molecules that the body can absorb. And trypsin, chymotrypsin, carbopeptides. I'm going to say all these just because I want to. Pancreatic amylase, lipases, disaccharides, and dipeptidases. All of these things, and I did look this up, that peptides aid in the breaking down of proteins. So these are secreted from the small intestine that are going to help break down proteins within the small intestine. So all of these things are aiding in digestion, and they're breaking down carbs, fats, and proteins into molecules that monogastrics can use throughout their body. Now, that's all we're going to go through today. We have a video that we're going to watch. I want to be able to post this video on YouTube, and if I play this YouTube video, it won't allow me to do that. So I'm going to stop the recording here shortly. I need everyone to stick around. You need to watch this video. They do a nice job of, these English women do a very nice job of talking about and demonstrating what the horse's digestive system looks like. With a monogastric, but it's a little different than pigs and humans in that they have a very active and functional cecum. So we're going to learn a little bit about that, and we're going to watch that. But I need to stop the recording in order to watch that. I will post this video for those who missed class today so that you can go back and watch it. But for now, we're going to stop the recording and jump into this video.